is identify the given histological slide. So here you have to identify the slide. If you look at the slide, then you can make it like from the first appearance you can tell that the central part is here and the peripheral one. First of all, if you remember the basic ones, let's make it from the beginning. That one thing is very clear, that it is the hematoxin and eosin stain, HNE stain. Okay, and in HNE stain, what happened that this one, let me just take one cell here, like, or you can take this one. So this one is the like pink color, and their central one is blue color. Blue color identify the nuclear material or nucleus, and the pink color is basically stand for the cytoplasm. Now cytoplasmic and nuclear one, and the this one which has the basically like here you can see the white color or the white zone, they are the chromophobic, or they are the chromophobic. So these can be the lipid one, these can be, or you can say the fat or simple, like in foreign or exogenous. Okay, so this was a basic concept. Now looking at the diagram. Now how to make it answer that what is given in the diagram. So the central part is here. Central part is basically paler in color, and the outer one is having a lots of different structures. So in the given structure, what does it mean and what does it contain? So this is the this part, this zone. This one is the very famous in the pancreas, which is also known as islets of Langerhans. And the surrounding one, that there is because it makes the endocrine part, and the surrounding one, they are the secretory cells, so these are the basically exocrine part of pancreas. Exocrine part. So here it is the acini of pancreas, you can say, to make it so we can say like it is the acini of the pancreas. Acini of pancreas. Now in the given structure, we have the two part, islets of Langerhans and the exocrine part, that is acini of the pancreas. Now islets of the characterized by the secretory cells that are supported by the reticulin fibers and containing the numerous fenestrated capillaries. So here are the fenestrated capillaries are present, and because they are too small, and in HNE stain it is not possible to look into the deep one. So these are the delicate capsules, and around each islet are the paler and surrounding exocrine cells, and they have the less rough cytoplasm reticulum. Now the given image is having here islets of Langerhans, and the outside there is a pancreatic exocrine part, exocrine cell. Now what happened that to go one step deeper, what we can say that they are the basically in islets of Langerhans, there is alpha, beta, delta, and pancreatic polypeptide cells. Out of which the responsible things: alpha for the glucagon, beta for the insulin, delta responsible for the somatostatin, and polypeptide, these are the pancreatic polypeptide, have the several gastrointestinal effect. Now okay, so to be clear with this image, you know that it is the pancreas one, and now to go one step deeper, to make it to look at the options, how other options look like. The first option here, it is a lymph node. What is a lymph node? Lymph node is it is the secondary organ, or you can say it is a secondary lymphatic organ. So how it looks? So to be very clear, from the beginning, that lymph nodes are the secondary lymphoid organs. Actually, these are the secondary lymphoid organs. What are the primary lymphoid organs? These are the thymus and bone marrow. Now after that, these lymph nodes, they have the structures of the nodes. Okay, because it word itself has a node. So now what are the characteristic feature of the node? So here this node consists of the, these are the basically, these structures are the node. So in the given structure, you can see that there is one blood vessel or the one lymphatic vessel which brings lymph, and like actually how does it look like? Actually, lymph node is something like this way. If it is a lymphatic system, so they have a nodular like of stay, like a temporary stay of the lymphatic cells, and that form the nodes, and that is the lymph nodes. Now in the lymph nodes, basically they have the one afferent, one efferent. So how it look like? When you look at the microscopic H&E stain, so what you will find out that there is one cortex part, and another one is a medullary part. Medullary is the central part. Now here the inner medulla, the cortex contains the aggregation of the V lymphocytes, the primary follicles, and the secondary follicles are the pale and central germinal layer. Now the inner medulla, now here the inner part is basically it has the consist of the medullary cord, the lymphocytes and plasma present, the inner one, and there is medullary sinuses between the cortex and medulla. There are there are ill-defined zone, so which is called the paracortex, so that contain the T lymphocyte. So here the given tubercles and germinal centers, and you can see that this one is a cortex part. So when you look at the image, if in the given image the dense blue color is split like this kind of a structure, it is the from the lymph node. Okay, now the next one, glomerulus. When we talk about the glomerulus, which comes in our mind, that glomerulus basically is present in the kidney. Now in the kidney, what we have that basically in the kidney, it's like this way. The glomerulus, this is by the light microscope. So in the glomerulus we have like basically they are supported by the mesengium. So glomerulus is a round, it is a round, and the second thing, it has the top of the anastomosis capillaries, and the glomerular capillaries wall is a filtering membrane consist of the fenestrated endothelium. So you can see that there is a one more structure is here like this way. It is very clear that it is not a single cell as in case of pancreas, but there is a group of cells you can see, because there's lots of like one complete structure is there. So here what happened that the glomerular tuft is supported by the mesengium and containing the mesangial cells of the non-cellular mesangial matrix. So in the given diagram, this one is the glomerulus, and this part is the PCT, proximal conjunctive tubule, and one part is their DCT, and in between them there is a collecting duct. So to correlate this diagram, it is actually from this part. We have the afferent arterioles, then Bowman capsule, then we have the efferent one. So the glomerular capsule is here. It is something like when we study. So this was the structure, loops of Henle, and then here is a collecting duct. So this part is PCT, proximal conjunctive tubule, and this is loop of Henle. This is the descending and this is ascending, and this is distal conjunctive tubule, and there is a collecting duct. Afferent arteriole and efferent arteriole, and here in between them there is a macula densa. So macula densa basically it is this part, macula densa part. Now here you can see the glomerular capillaries, loops, and thin delicates, and the endothelial mesangial cells are normal. Now after that the next option is here spleen. Spleen itself is a secondary lymphoid organ. Now in the secondary lymphoid organ, what you should remember that in the secondary lymphoid organ we have that there is a pulp. So to be clear from this part itself, it's a secondary lymphoid organ, or the lymphoid organ is a spleen. So here a spleen having here a splenic artery, or this one is the splenic vein. Now when we cut this part, like one small part of the spleen, and we'll look at them, so then the outer one is their red pulp, and the inner one there is it is not like it is white is in color, so it is a white pulp. And now one more specific to be like H&E stain, when we see, so we find that there is a red pulp, it looks like this way, and there is a white pulp, and this is the tubercle. So basically spleen has a structure like this. Okay, so spleen and lymph node, if you correlate, so you can find out it is almost similar, but in case of spleen there is a tubercle present. Okay, now after that The third option is yeah, it's pancreas. Now when we look at the pancreas, as I have explained you in the beginning, there's a body part and pancreatic artery, tail part. So you take one part of the pancreas, one simple sample, and here pancreatic islets, it is given in the diagram, and the acinar cells are surrounded by them, and their alpha, beta and delta cells, alpha for the glucagon, beta for the insulin, and delta cells for the somatostatin. Pancreatic blood cells are the fine minute vessels surrounded by the given structure. Okay, so hope this video will help you with the first explanation of the FMG question 2023. Now in the next question we will meet with the another question. Thank you.